All right, give me a minute to get everything set up really quick. Pull up my slides. And we'll get started probably. Why does this say 11:04 p.m.? Sorry, my time on my screen says 11:04 p.m. It is definitely not that. We'll probably get started just a couple minutes after 10, just to be sure we get our stragglers in. I don't anticipate having a lot of stragglers. Move this over here. Mhm. Let me use a pen. There we go. We'll try the subtitles thing. We'll see how that goes. To go let my dogs in. I will be right back. And we'll get started just a couple minutes after 10. I don't anticipate having many more people join. Why I can't see myself. Well, whatever. Let's just go ahead and get started. Here it is. Okay, so we're gonna go over what we talked about for week 3. And for week 3, what we went over was chapter 5. We talked about our eukaryotes. In chapter 7, we talked about our viruses. So we'll talk about our main points that we went over in these chapters, and then we'll go over the most missed topics for those chapters. All right, so starting with chapter 5. Chapter 5 for our eukaryotes. Remember, we've talked about the different organelles and structures within the eukaryotes. And so briefly, just the ones that we talked about for the organelles, we talked about the nucleus, which is the control center, or it contains the genetic information. So this is where the DNA is in the cell. We talked about the endoplasmic reticulum, or the ER. This is the site of big macromolecule synthesis, in particular protein synthesis. So which puts as our synthesis area. Actually, that was, does this erase? I do. Hold on. I have a new pen on this one. No. Yeah. Okay. Okay. So that's going to be synthesis of macromolecules. And so that'll be things like our proteins, our big chains of carbohydrates, our lipids, and then making those things. And then that will be moved into the Golgi apparatus. And the Golgi, that's the site of our, we will do some like modifications to the stuff we made in the ER, or like combining them together. We'll also be, my throat's scratchy this morning. We'll also be, this is our main site of packing and shipping. So shipping and transport here. All right, so that's the Golgi apparatus. This is just really bad the way that I wrote that. There we go. All right. And then we have, so that would be like step 1, 2, and 3 in making molecules. All right, so these are all the steps that are involved basically in our manufacture process in the cell. All right, and then we had the mitochondria. These are where we are going to be making energy for the cell. Spelling today is off. Really not wanting to cooperate. All right, and then we had the chloroplast. This was a site of photosynthesis. Right, and then we had the vesicles, right? So our 2 vesicle types that we talked about that are having specific functions in the cell, our vesicles that we talked about are the vacuole and the lysosome. The lysosome, so the vacuole is the like basically for storage of water and wastes and other components of the cell storage unit. And then the lysosome, it also stores, but it stores something very specific. And the thing that it stores is going to be the digestive enzymes, as well as looking at the closed caption, and the closed caption was throwing me off. The thing that it stores is going to be digestive enzymes to break down bad guys, waste, things like that. All right. So that's the gist of our membrane-bound organelles. We also still have things like ribosomes and those sorts of things that have functions within the cell, but these guys are unique to eukaryotes. We also can have cell walls in some cases. Remember that fungi and algae will have cell walls. but that the other guys won't. Okay, so I could just automatically just clear everything. There we go. Okay. All right, so the next thing is our endosymbiotic theory. This is the idea that some of our membrane-bound organelles, like our mitochondria, here we go. This is why I'm not seeing myself. So our mitochondria and our chloroplasts, these guys have their own circular DNA. So remember, normal eukaryotic cells, their DNA is linear. It comes in chromosomes that are linear. With bacteria, they have one big circle of DNA, and that's their whole genome. So our mitochondria, or organisms that have chloroplasts, like our algae and our plants, those organelles have their own circular DNA. They have ribosomes that look like bacterial ribosomes. They divide independently of the cell. Right? And they have double membrane. And this is just some of the evidence that we have that supports the idea that maybe what happened back in the day when we were evolving is that we had a cell that ate up another cell, and then those developed a symbiotic relationship, and they were probably both benefiting from it, which we call mutualism. And that mutualistic beneficial relationship that they formed became a permanent one, and eventually they became our organelles as our mitochondria and our chloroplasts. That whole situation, we can summarize that basically as mitochondria and chloroplasts. There's evidence supporting that they evolved from bacteria that became permanently depended upon by the host cell, and now they're organelles, right? So that's basically the idea of endosymbiotic theory. Right, the next thing was mitosis. Briefly, because I don't want to go over every single step of mitosis again, but briefly, with mitosis, our steps for mitosis were PMAT, right? P was our prophase. This is when we are preparing. So this is our prep phase. We are just condensing down our DNA. We're making structures that are going to be involved in separating our cells into daughter cells, that stuff. That's our prep phase. M is metaphase. And this is when we're lining up our chromosomes in the middle. So literally your cell would look like this. And all that's going to be lined up right down the middle of the cell. Anaphase. This is going to be pulling those apart. So your cell would look like this, where you would have those X's being pulled apart now into single parts. And so these are the things that be pulling them, called spindle fibers. We'll pull them apart from one another. And then in T, this is telophase. And I say this is the phase where you are totally done, or at least making preparations to be totally done. So this is where we will have our individual chromosomes that we've made on each of the daughter cells. So these are our chromosomes, our X's that we pulled apart, right? And they would be in our individual daughter cells. We'll be reforming our nucleus around them. So that's the nucleus. And now here we have our daughter cells, and we'll be pinching off that membrane in the middle to make those. So that will be the example of the very end of telophase. So those are the stages of mitosis. Remember, mitosis is the process that we are going to be using to make identical daughter cells. So it's going from one cell to two identical daughter cells. The process that we use to make our sex cells is called meiosis, and it's spelled like this. That's how we're going to make our sex cells, our gametes. All right. All right, we talked about fungi and their structures. The fungi, I'm not going to go over that too much, but the things that we do want to remember, first of all, one, that yeast are fungi, that the branching structures that we'll see are called hyphae, that the entire vegetative body, you guys remember what vegetative means, right? So vegetative means that we're like active, multiplying. That word vegetative is a descriptive word that can also be applied to any situation where we would be active and multiplying. It also happens to just be an official word for any bacteria that are active and multiplying. And remember, bacteria that are dormant would be called, in our inert state, would be called the endospores, right? So the ones that are hunkering down for the good. So those structures are called endospores. But anytime that we see cells that are active and multiplying, our entire vegetative form, our entire what do you want to call it, main body or entire body, all together. So here's our points. I'm going to make points so we can separate where my sentences are going. So that is called a mycelium. And so the whole reason I'm putting in this much detail here is because when we look at things like mushrooms, the mycelium look like roots to us, but that's actually the main body of the fungus. The mushrooms are just the fruiting body or the reproductive structures that they have. So that is the difference there. When we see mushrooms, we think that's the fungus. Not, maybe if there were roots under there, that's roots. That is absolutely incorrect. It's not like plants where the main part that you see of the tree, that's the tree and that's the main body, and that's where a lot of the action is going to be happening, is in like the leaves getting photosynthesis, and all this action's happening in the tree, and the roots are just there to absorb nutrients from the soil. But that's not the case with fungi. Fungi don't have roots, they have mycelium, and the mycelium is the actual functioning body of that fungus. And then mushrooms are literally just the reproductive structures. Okay, so mushrooms are just reproductive structures, and so that's why I'm bringing that up. With our molds and our yeast, they don't have a separate structure outside of what you see. So that molding, branching body of the hyphae and everything, that whole thing is their mycelium, right? And so it's pretty straightforward with the molds and the yeast. The yeast, its whole structure, that's basically what we're going to be talking about. But yes, so that whole main functional body altogether is called the mycelium. Remember that mushrooms they're going to have their reproductive structures. That's the actual mushroom that you see. Okay. All right. And then for reproduction, yes, they can just spread out. So they have the ability to spread. They have ability to fragment. So that just means like you could break off a piece of mold and put it on a new like piece of bread or a new strawberry or whatever it is, and it'll just grow there. That's fragmentation. But these guys also can reproduce sexually. So that means we can get some genetic exchange going on. We didn't talk about the details of that. But their main form of reproduction is going to be asexual. And their asexual structures of reproduction were our sporangiophores or sporangiospores. So that would be the actual spores that were made. So I'm going to put sporangio, okay? The sporangio related structures or the conidio related structures. Okay, so sporangiophores are the structures overall. Sporangiospores are the spores that are being made by the structures, and sporangia are the organisms that make all this. That's why I'm just going to say focus on sporangio. I will not make you distinguish between sporangiospore or sporangiophore. I'm not going to make you distinguish between those. Okay. Sporangio guys, these guys have a covered structure. So they might have something that looks like this, and all the little spores that they are making would be within a cover. And they'll just burst out. So the conidio guys are uncovered. There's definitely a V. Okay. All right, so the sporangio are covered, the conidio are uncovered, and what the conidios would look like would be like this. And they would literally be like the same thing, just no cover. And they'll just release their spores freely. That's the difference between those guys. All right, moving on. The protista, the algae, and the protozoa. Algae are photosynthetic. I don't stop popping up with a thing. You guys aren't seeing the window that it keeps popping up with, but it keeps popping up with a thing. I'm getting used to this new tablet thing, so it's taking me a little while to get used to that. Algae are photosynthetic, and protozoa are always single cell. All right, so for our algae, we're not really going to talk about them too much. I didn't mean to draw a line through it, but hey, there it is. We're not really going to talk about them too much because they aren't going to really be that important for us medically, as far as what we're going to be focusing on for our class is the medical aspect of these guys. But protozoa, we will be. So there's 4 different groups of protozoa. 1, 2, 3, 4, and they're all grouped based on how they move, like their motility or whether they are modal. So our first group, they move by what we call pseudopods, or fake feet. Pseudo, fake, podia, feet. So pseudopods, these are our amoebas. All right, our second group, these guys move by flagella. Our third group, these guys move by cilia, which are those tiny oar-like appendages that filter their environment or can help the organism actually be modal. And then our fourth group, these guys are our non-modal group. And literally all of our non-modal group are parasites. And it says modal. That is not the correct word. And that occasionally I'm catching some of the incorrect stuff. And there's some motile is what we're talking about. Motile. Okay. The pop-up screen is killing me here. I'm really trying. Come on, guy. Give me my eraser. Where'd my eraser go? I don't know how my eraser is not working, but it isn't. Sometimes it works and sometimes it doesn't. Sorry. Anyways, so I can't get it to work. Anyway, so that's it for those guys and how we're going to be organizing them. We'll talk a lot about the diseases that these guys could potentially cause when we get into later chapters. And so these guys can cause all sorts of diseases, but a lot of them are associated with diarrhea. So I'm just going to warn you guys about that when we get to that in chapter 23. But we just need to understand the basics of our organisms right now. So that's fine for us for now for those guys. Moving on to our helmets. I said erase everything. My guy, it's still there. And that doesn't give me the option to erase it. So we have to have that there now. So our helmets, these guys are our parasitic worms. All right. And so our platyhelminthes, these are our flatworms. It went away. Flatworms, and our cestodes are our tapeworms. Our trematodes are our flukes. And our nematodes are our roundworms. All right. So that is the gist of those guys. So we have within our parasitic worms, we have our flatworms and our roundworms. Our flatworms can be subdivided further into our tapeworms, which are a type of flatworm, and our flukes, which are a type of flatworm. Okay, so be aware of all of those terms that are associated with those helminths. All right, moving on. Chapter 7. So for chapter 7, our main concepts, and again, I'm not going to break these down in as much detail because I feel like the detail's kind of here, but viruses are obligate intracellular parasites. It's actually really important that we remember that about these guys. Okay. So they are obligated, they have to do this. They have to be intracellular, inside of cells, parasites. So causing harm or damage or using up the resources of a host somehow. So this is a negative relationship for the host, but the parasite itself, the virus, is benefiting. So that's a parasitic relationship. All right. So our viruses, an individual active virus is called a virion, and a single virion consists of they always have a genome. It's really important that we're remembering this. They always have a genome, and they always have a protective protein coating called a capsid. Okay, so they always have those two things. Always have those two things at the bare minimum. There's a question on one of the quizzes that maybe I had one question from one student, so I know that there are more than one student that are getting this wrong and asking and wondering why they got it wrong. Does every single virus have DNA? No. They have a genome, they have genes, but their genes are not always DNA. Okay, sometimes some viruses have RNA for their genome. This is different than cells. Cells always have DNA, and it's always double-stranded DNA. But viruses can have 6 different genomes that could be DNA or RNA. Their DNA, it could be double-stranded. It could be positive sense single-stranded, which means that if a ribosome could read DNA, which it can't, but if it could, that it would be able to read that positive sense version and make a protein out of it. Whereas the negative sense, it would need to make a positive copy of it, and then it could read the positive copy. Okay. So ribosomes only read single-stranded RNA, but they could also have double-stranded RNA. They could have positive single-stranded RNA or negative single-stranded RNA. So you can imagine if you were a positive single-strand RNA viral genome, a ribosome can attach to that positive sense single-stranded RNA genome and just start cranking out protein products. We don't need to go from DNA to RNA to protein, which is our usual steps in a cell. Stop. All right. And this process here, because we're going to mention it in a moment, this process from DNA to RNA is known as transcription. And this process here is known as translation. And this is done by the ribosome. And this is how we always thought that every single living thing would have to do their genetics, go from DNA to RNA to protein. But viruses disproved that in a lot of different ways, clearly. So they can have all these different kinds of genomes. So there was a question on the quiz that asked basically like, what do viruses have to have? They do not have to have DNA. And that was one of the choices. And that was a very popular choice on the quiz. They do have to have a capsid. All of them have a capsid. All of them do. Okay. So they have to have a genome, but that genome does not have to be DNA. They have to have a genome, but their genome does not have to be DNA. They have to have a capsid coating made out of protein, period. All right, so these are the bare minimums. You will also have spikes that help you get into the cells. Now, the reason I don't have they always have spikes because these are technically not separate from the capsid. It can be part of the capsid. So this gets into that gray area. But all of the viruses will have spikes as well, but it could be part of their capsid, or it could be something that's on their envelope, which not all viruses have. So if you have an envelope, this is a stolen membrane. It's stolen from the host cell. I don't understand why this thing erases things while I'm writing it. Oh my gosh. Oh my God. Anyways, so I was about to write membrane, so we can pretend like I wrote membrane, please. This is getting so frustrating. Anyways, stolen membrane. Right, and stolen from the host cell as it is like pushing its way out, it will like basically wrap itself up with the membrane as it is leaving the host cell. So that's where it gets that envelope from. So a viral envelope is something that only some viruses have, and we would call you enveloped if you have an envelope. If you are only consisting of a capsid and a genome, and you don't have an envelope, if you are non-enveloped, you're called naked. All right, there. All right, animal viral infections. These guys go through the following 6 steps. Adsorption, this is where we're going to be attaching to something. Penetration, this is where we're going to enter. Uncoat, this is where we're going to drop that capsid. So we're going to say remove capsid. Synthesis, we're going to make copies of our parts. We're going to make parts. Assembly, there should be a comma here. Assembly, this is where we're going to put parts together. I'm having a good time, I promise. All right, and then release, whenever we are releasing active virions from the cell. So we'll say viruses leave cell. All right. So those are the 6 steps in viral animal cell infection. Then we have the various cytopathic effects. These are changes that we would be able to see at the microscopic level that are happening to cells. And I want to be very clear about this. Viruses are so small, you will not be able to see them with a typical microscope. You cannot see viruses with a light microscope. You have to use electron microscopes, and you can't just look at cell samples with electron microscopes. They take very intensive and very complicated preparation to prepare a sample for electron microscopy. And for example, we don't even have an electron microscope on our campus. So they're incredibly expensive and they're incredibly hard to use. So they require special kinds of training and things like that. So compared to a light microscope anyways. So those guys are basically something that you don't just use that to look at cells typically. And we don't typically use electron micrographs for like diagnostics. But if I get a tissue sample from a patient, or I'm able to get a swab that has samples from a patient, and then I can look at the cells in that sample, those cells might look like they have something going wrong with them. And those are the cytopathic effects that we see from viral infection. And we talked about examples of some of those cytopathic effects, like the syncytia formation, when the cells next to each other start to merge together because those viral proteins act as keys to enter the cell next to them. If you put those on the outside of your own infected cells, now your infected cell can merge with the cell next to it. And so it merges cells together, creating these giant multinucleated, what used to be multiple cells, making giant cells out of them. And that's called syncytia. And then we could also see cells detaching from surfaces, cells not being oriented in the correct orientation in regards to their environment and how they're interacting with the cells nearby. And in multicellular creatures, that can have a massive effect on how it's affecting the organism. And so those are called cytopathic effects. All right, bacteriophages, that's a term referring to viruses that infect bacterial cells. This thing is driving me nuts. I wish I could change that setting so it stops popping up with that thing and erasing what I'm doing. Bacteriophages are viruses that infect bacteria, and they only infect one species of bacteria. And this is going to be important later on because we can actually use this for diagnosis. If I am able to have a sample from my patient's diarrhea that I was able to culture, and if you don't think that that's something that happens, I hate to break it to you, that's something that happens in the lab. We might be culturing diarrhea bacteria. And so you might want to know what's causing that diarrhea. We can try to infect it with a phage that only infects salmonella. And if it infects your patient's sample, guess what? Your patient has salmonella. And so we can use that as a diagnostic tool. That's called phage typing. I'm not going to test you about that on this test. I want to be clear. It's just an example of why we might want to know that. So bacteriophages only infect one species of bacteria. They don't infect any other species. And they're always, when they make copies of the virus, they are always going to lyse cells open. Okay, so what does that mean? That means we're going to break out of our host cell, not by budding out and covering us with a little blanket of membrane, not by having the cell push us out, but by literally bursting the cell open. Anytime you burst a cell open, that's called lysis. That can happen because viruses are bursting out, or it could happen because too much water is getting in the cell, etc. So that's how bacteriophages will work. They can go through two phases, bacteriophages. So I'm going to put phages, which is viruses that infect bacteria. Okay. So phages can go through two types of life cycles. The first phage, the first phase of phage life cycle we'll call the lytic cycle. Now I want you to erase what I'm writing, my guy. There we go. Okay. I can't today. Lytic. In the lytic cycle, we are making copies of the virus, copies of the phage, and then we will also be lysing the host cell to get out. We're just going to burst out of it. So this basically this is our like an active phase, right? We're making copies of the virus. And that's typically what viruses are there to do, right? Reproduce. Our second phase of phage life cycles can be, and not all phages do this, okay? This is called lysogeny. Oh my God, this is killing me. Lysogeny, okay? All right, so this part, the lysogenic phase or lysogeny. The gen is what I want you to focus on because this is when the phage genes insert into the bacterial genome. Okay. And so it becomes part of it, just like we talked about with HIV and those things in our human stuff. So when that happens, that viral genome is called the prophage. When this happens in humans, it's called the provirus. So this is analogous to a provirus in animal cells. This is a P, okay. Provirus, so in animal cells. It says genome. I know it's an M, but it's killing me with this thing erasing what I'm trying to write. So when viral DNA gets incorporated in the host DNA in animal cells, it's called the provirus. When it is a phage, a bacterial phage, which is a virus that infects bacteria, and what is that? Viruses that infect bacteria, then it's called a prophage. Okay, it's the same situation, incorporating the DNA into the host genome. In the case of animal cells, provirus. In the case of bacterial cells, prophage. Okay, and so that process where we can switch to incorporating a prophage into the bacterial genome, that is called lysogeny. And that's what that's called. Okay, so be able to hide out like that. So that's a hiding kind of a thing to do. And this can last forever, basically. They don't have to ever make active copies of each other unless they want it to get to, to spread their viruses. And so that can depend on environment. There can be other triggers that can induce that, but you have to go through induction in order to get you to switch between the two phases. Okay. So viruses, phages, phages that can switch between these are called temperate. Think of somebody that has a temper, being able to switch between having the temper and not having the temper. So switching between being lytic and doing normal virus stuff or being hide out and just wait kind of virus stuff. Okay. So those are the differences that we have there. And that is basically the summation of the two phases of bacteriophages, the lytic and the lysogenic. Okay. So we already mentioned our provirus. We've already gone through all these things. Just keep me on track. The oncoviruses are viruses that can cause cancer. So things like HPV, they can create proteins that disrupt the cell cycle. They can actually incorporate themselves into your genome, and just by doing that can disrupt your cell cycle. Everything that we have as checkpoints to make sure that our cells aren't growing out of control, that's part of our cell cycle. And controlling whether we're going through mitosis and things like that, we have to be able to have checkpoints for that. Otherwise, cell growth can go unchecked and then you have cancer. And so viruses can affect that system and lead to cancer development. So that's oncoviruses that are able to do that. We think about 13% of all cancers might be caused by viruses. It's probably more than that, but that's what we have evidence for so far. All right. So now that brings us to our prions. Prions are misfolded proteins. So if your normal protein looks like this, where red equals hydrophilic or water loving, right? I guess it's the Super Bowl this Sunday. Is it this Sunday or next Sunday? I don't know. Super Bowl's coming up on a Sunday. Someone asked me what team I was rooting for. Obviously, I don't know what's going on with that, and I'm not tracking it, but I have been informed that the Eagles are playing, and so I don't know who else is, but I know who the Eagles are, and I went to school in Philadelphia. I lived there for a while, so I'm going to go with the Eagles. I'm just saying. Okay, somebody asked. All right, so you might have, if we have red hydrophilic or water loving, and then we will put, I don't know, purple. For our purposes, purple will be hydrophobic or water hating or water fearing. So it does not like water. Okay. So this is a protein. This is one single protein, and these are just parts of that protein. Okay. And so if this is misfolded, I swear that this is just going to drive me up a wall in a moment. Okay. If this is misfolded, and let's say now it looks like this. So it's kind of flipped its like orientation of what's inside and what's outside. If that's everything that's going on outside of that is watery, like our cells and most of our body, well, the purple part doesn't like being around the water or the cells or the contents of the cells or anything like that. So if your protein becomes misfolded, and now we've got hydrophobic on the outside, our non-polar groups on the outside, now these guys, those polar stuff, will stick to each other, and they will basically hide out in, like, they'll stick together. So these guys will all stick together to try to hide away from the water. And yeah, I mean, they've got their other groups on the inside, but that's not interacting with the outside. That's all hidden. So now we can actually contact our healthy proteins with the red on the outside and cause them to fold up all nice and ridiculous like this. I'm going to kill this computer. Okay. So we can actually cause our normal to turn into our misfolded. And then those misfolded guys eventually will get more and more and more of them, and then they'll stick together, and then those will create holes in the brain tissue where this would be happening. And it makes your brain look like a sponge. So spongy form, the form of the brain is a sponge. Encephalo means brain. I need to like work on the nuances of this tablet because it is killing me. Brain, pathy means disease. All right. So that's the idea with that. I'm just trying to write an S. Anyways, so you get it. Okay. Let's move on. So these are just misfolded proteins. So if I ask you, what kind of genome do prions have? So prions, what kind of genome do they have? No is the answer. They do not have a genome. They're literally just misfolded proteins. Your body is coding for proteins that are normal and they just aren't folding right. Why? We don't know. Okay, that's science. Science doesn't have an answer for that yet. So they are literally just misfolded proteins that will cause other normal proteins to also misfold. And then that just causes a cascade effect. And then they build up in your brain and cause holes, and then you die. It's 100% fatal. There's nothing we can do for you. It's incredibly difficult to diagnose. In fact, there is no 100% diagnosis until after you're dead and we can look at your brain tissue and remove it from your skull. We can do some testing on CSF from people that we suspect might have this issue. So we can take cerebrospinal fluid and send it off to specialized laboratories that test for this. There's no testing for this in Oklahoma. So you'd have to send this to like the CDC or to Los Alamos, where they are doing these testing on these proteins, because this is an incredibly rare disease. We don't test for this very often. And you're not going to have symptoms until 30 years later. So good luck. All right, so we're going to move on. For chapter 5, our first most missed topic will be our organelles. Knowing what our organelles do in our cells, what they're called, and their functions, and everything like that. So our major organelles were our nucleus. This is your genetic info. Our endoplasmic reticulum, or our ER. This is where we're going to be making proteins and other large molecules. If it's making a protein, it uses ribosomes that are going to be stuck all over that endoplasmic reticulum membrane and making it the rough ER. So the rough ER is where we're going to have proteins made. The smooth ER is where we're going to be having other things made. The Golgi apparatus, this is packing and shipping and creating vesicles full of product that we can move throughout the cell or outside of the cell. I'm having some lady cramps today. All right, we've got mitochondria. This thing, mitochondria. These are the powerhouse of the cell. These guys are going to make all the energy that the cell needs in order for it to perform anything, pretty much ever. And so that's usually going to be in the form of ATP. That's our energy molecule. And then we have chloroplasts. Chloroplasts are where we're going to have photosynthesis in our organisms that are able to perform photosynthesis. These are only going to be present in plants and algae. I am going to lose algae. All right, so how are we going to remember this? Never eat grandma cookies. Never eat grandma Mama's cookies. So now we know our 5 major organelles, and now it's on you to remember what they do, right? All right, so our example questions. In a laboratory experiment, a eukaryotic cell is exposed to a chemical that disrupts the integrity of the mitochondrial membrane. So as we're reading this, we have a chemical that causes your mitochondrial membrane to be disrupted. What does the mitochondria do? It makes our energy. We put mitochondria makes energy. I'll put E like this so we know that's energy, okay? So we remember what it does. And so if we're disrupting our membrane for the organelle that makes our energy, we're probably going to disrupt that energy making process, right? So that's probably what we're going to get at here. After exposure to that chemical, the cell shows a significant drop in ATP levels. Oh, I bet it does, because the mitochondria makes our energy. So now we're not making ATP, which is our energy molecule, and we have an increased oxidative stress. So oxidative stress, if you remember from chemistry, this is referring to redox reactions. Don't you love it? Reduction and oxidation. So we're talking about issues dealing with electron transfer in molecules. Now, you don't even need to know what that means, okay? So we're just going to leave that out. We didn't talk about that in our class yet. That's a chapter 10 thing. So we're just going to say, cool, I don't know what that is, but I know that the mitochondria makes energy in the form of ATP. So we're just going to stick with that for now. Which of the following cellular functions is most likely impaired as a result of this chemical treatment? So this question has given us complicated information that we might not yet have covered in our class, but we will eventually. But it's given us information that basically is asking us, if we put a chemical on our cell, our mitochondria aren't working right anymore. What's happening? That's what the question's asking. Put a chemical on the cell, mitochondria aren't working right. What's going on? So is our protein folding and modification being disrupted in our endoplasmic reticulum? This doesn't have anything to do with that. Are we having intracellular digestion from molecules from our lysosomes? Are our lysosomes being affected? How would that be happening? Is our energy production through aerobic respiration in the mitochondria being affected? Yeah, energy production in our mitochondria is definitely being affected here. So yeah. Are we having issues making lipids and steroids in our smooth ER? No, we're affecting the mitochondria, okay? So you guys would probably be seeing a question like this on the test, which again, my questions on the test won't have this level of complication. I'm giving you harder questions on these from time to time so that I can get you guys thinking about it a little bit more complexly. The questions on the test should not be this hard for you. If the questions on the test feel this hard for you, that's a you problem. You didn't study well enough, because I'm not writing my questions on my test this hard, okay? But this has all the information that we needed to answer it. You just need to see through the weeds of the complicated language. Guaranteed that 90% of you that would be taking my test, if I put this question on the test, would just panic and your brains would stop working and they wouldn't think to answer the question for you. Tell me I'm wrong. Tell me, because this is usually the type of thing that you'll read this question and your brain will see the words and maybe understand the words individually, but processing what it's asking and getting to that point of figuring out how to break down these things that have all these complicated situations in them, most of you guys, your brains will just shut down. And then you're left to just try to figure out how to even guess from this. And that is a panic reaction that you have to practice to get over, because when I am telling you guys, it doesn't get easier from this class. This is not the hardest class you're going to take, period. You have to learn to overcome that panic response. So start working on that. Start teaching your brain that it doesn't have to panic just from seeing some words. And tell me I'm wrong. If this was on a test, right? Next question. A research team is studying a eukaryotic cell, so it has organelles and a nucleus, we know that automatically, that shows a marked decrease in the ability to synthesize proteins. Where does protein synthesis occur? The endoplasmic reticulum. So we're going to start thinking about that. The endoplasmic reticulum. What things make proteins? ribosomes, right? So we're going to start thinking about those. All right, they observe that the cell's rough endoplasmic reticulum, so that's the one that's studied with ribosomes that makes proteins, that its ER looks damaged and its ribosomal RNA levels, the things that make up the ribosomes, are low. So our cell has been seen that it can't make proteins well. Our ER is messed up and we don't have ribosomes really. What conclusions can we make that are accurate regarding our cellular functions? Okay, so our ER is messed up, our ribosomes are messed up, we're not making proteins right. What's going on? That's what the question is. Okay, so our choices are, we have a decrease in protein synthesis because the Golgi is malfunctioning, which is responsible for protein modification and packaging. The Golgi is responsible for protein modification and packaging. Do we have any evidence in our question that suggests that that's the problem? We do not, so it's not this one. The damaged rough ER, all right, the site of protein synthesis, and the low ribosomal RNA levels, so therefore low ribosomal function. So our damaged ER and the ribosomes suggest that the protein synthesis is impaired because the rough ER is the site of ribosome attachment and initial protein synthesis. That's literally what we have been saying leading up to this. So this is probably our choice. The malfunction of the mitochondria, okay, so we've got mitochondria now, is causing decreased ATP production, which is affecting protein synthesis by reducing cell energy supply. If we had disrupted mitochondria and we couldn't make ATP, would that affect our protein synthesis? I mean, I would ask you guys to answer, but I know you're not going to. Yes, you need energy to do anything ever. If you're doing a thing as a cell, you need energy for that. It doesn't happen spontaneously. So if we did mess up our mitochondria, it would affect protein synthesis. It would also affect everything else in the cell that the cell was trying to do. But does the evidence that we're given in the question suggest that the mitochondria is the problem? No, it suggests that the ER is the problem. So C is not our answer. Even though that would affect protein synthesis too, it's not the answer. And if you were looking at this being like, obviously, this isn't the answer because mitochondria isn't the problem. Well, yeah, you're right about that. But do you realize that affecting your mitochondria would affect your protein synthesis? Because if you didn't make that connection, you're not understanding the material well. Right? So this is what I'm getting at here. You need to see the big picture stuff and how these things are all interconnecting because questions on the test will demand that of you. So the reduced activity of the lysosomes are leading to undigested proteins, and then those undigested, basically lysosomes contain stuff that can break down anything. Are we releasing the digestive enzymes and breaking down the proteins, and that's what's having the effect? That's not the problem. We've already talked about what seems to be the problem, and that's the endoplasmic reticulum, and that's why B is the answer. Okay, so we've got a lot of critical thinking going on in these questions. It's not just about saying, we've talked about the ER, so the ER is the answer. If you are just looking at these questions like that and not understanding what the other stuff is going on in the background, you will not do well on the test. Okay, next thing, I can't drive that home enough. So many people just think that it's going to be all just happy, find the answer, like match the term to the definition. And there will be questions like that on the test. A lot of the questions will all be on the test. A lot of the questions that are on the test will be pretty straightforward, definition to term and basic questions like what we have in the quizzes and applying that stuff. If you're having trouble with the questions that are on the quizzes and you're relying a lot on looking up the answers in your notes to answer the quiz questions, those are the easy questions on my test. I have questions that are on my test that are like those, and I consider those to be my easy questions on my test. So keep in mind, if you're having issues with the quizzes where you are relying very heavily on your notes to get you through those questions, that's a sign that you need to actually take time to understand the material outside of relying on your notes to get you through it, because you aren't allowed to use your notes on the test. Okay, test is next week. Somebody asked me what my average on my first test usually is. About a 60 is my average. Prepare for that. Okay, that's why it's the lowest score. That's why it's the lowest of all my tests. It's not that the other tests get better scores. So let me put it this way. People do terribly on my first tests. I have a chat. Hold on, let me put this up here. Okay, I've gone over like the test setup probably like 20 to 40 times already. But yes, we have 50 multiple choice questions on the test. A handful of those questions will be critical thinking questions. I say a handful. I'm not going to tell you exactly how many. I feel like everybody should be studying for that test for every question to be a critical thinking question. Don't slack on your studying because you think only a couple of the questions will be critical thinking. Because I'm telling you, only a handful of them are critical thinking questions. Most of them will be questions similar to what we see in the Kahoots and the quizzes, and people still get a 60 average on this test. It's next week on Wednesday or Thursday. So if you guys are ever wanting to know about when your tests are going to be, or when you want to keep up with when we're doing reviews that are official, that are in class, like our presentation days or a test like that, that's all on our syllabus schedule as well. They're going to be in class. The test is in class, in person. You have to bring your laptop or your tablet with you, and we'll be doing it on Lockdown Browser. I do not use ProctorView in my class. Number one, I think ProctorView puts undue stress on you guys, having strangers watching you and getting in trouble for just having a book on the shelf in the background that's about like Cat in the Hat or something. And then on top of that, I think that there is a massive opportunity for cheating on ProctorView tests. So I don't do ProctorView. I do my tests in person. So be prepared for that. All right. So 50 questions, multiple choice. A handful of them will be critical thinking test questions. There will also be plenty of questions that are like basic, like what is the mitochondria? And you tell me that the mitochondria makes energy for the cell. Basic kind of term-based questions. And then there will be questions kind of in between with a little mix of that stuff. Which is some of the stuff that like what we have seen a little bit of in the quizzes, right? So there's a little bit of critical thinking stuff type. I want to say critical thinking, that's not accurate, but it requires a little bit more application than just a definition and a term, right? So applying that stuff. So we'll have a mixture of those types of questions on that exam. 50 questions, multiple choice. Most of my students take 30 minutes to earn their 60% on it. Okay. So I'm being honest with you guys. And that's why my test is not written in a way to trick you guys. I have warned you guys time and time again that you will not see the same questions as you've seen previously. Typically, you might have, like I said, 3 or 4 questions that you had on your quiz. I'm not going to tell you which ones. All right, some of them might be the same. You'll have critical thinking questions, none of which you have seen. You will have a handful of those, like 5 or so. You will have questions that are kind of in between that require a little application, but don't have heavy critical thinking aspects to them. Some of them will be similar to what you have seen on the quizzes and the Kahoots. All right. 50 questions, multiple choice, right? And I guarantee that if you are preparing using the study guide, and I can't stress this enough, look at the study guide. If I were to pick up that study guide, and this is images that I can take, like, let's say I took this straight from our study guide, because it's pretty similar to like this stuff. This is not from the study guide, but it's derived from it. And I were to say on the study guide, one of the things on there that I have is I describe to you what a cyst and a trophozoite is. So if I were to your face to ask you, instead of giving you multiple choice questions, instead I were to your face, what is a cyst? What's a cyst? Explain that to me. What's a cyst? If you can't actually put that into your own words and explain to me a cyst, and also if you're explaining to me a cyst, you'll have to explain to me what a trophozoite is and what a protozoan is. And probably also what, if you're talking about protozoa, be able to explain to me what a eukaryote is and all this stuff that we've learned about in the course that all relates directly to what's in your study sheet. If you can't explain those things to me and derive those relatedness between those things in your own words without referring to your notes, you are not prepared for my exams. You need to do that with the entire study sheet. period. And I've said this multiple times. Going over questions and going over questions and going over questions and answers repeatedly, only ever getting that same answer. I got it right again. I got it right again. I got it right again. I got it right again is not going to help you with understanding. I hate to break it to you. That is a terrible way to study. Repeating the same questions and getting the same answers over and over and over again, just because they're right, doesn't actually help you understand the material. It just helps you get the same answer every time. So if you've already answered a question correctly, try rewording it. Try flipping the answer into a question. So if your question asks you, what is the active feeding stage of life for a protozoan? And the answer was trophozoite. Rewrite it as, what is a trophozoite? And the answer would then be one of your choices, and multiple choice might be, oh, it is the form of a protozoa that is the active, metabolically active, feeding, multiplying version. Rewrite these in your own forms. Have ChatGPT rewrite questions for you. Have ChatGPT. One of my students took ChatGPT, took my critical thinking questions, took the study sheet and said, here, ChatGPT, here is the content that I have to know for Unit 1. Is the study sheet. Here are some questions that my professor gave me that are critical thinking questions. Write me more. And it did. And she is using that as a method to study. Excellent. Excellent work to that student. Those of you who are just going over and over and over the same questions with the same answers on Quizlet, which is just flashcards, it is. Come at me, okay? Quizlet's just flashcards. If you're just going over the same questions and answers and you don't even know why the answer is right, if you don't even know why the other answers are wrong, that is not going to prepare you well. You might pass it. You might get a C, and you will not get an A, that's for sure. So you need to understand this stuff, and I can't explain it any better than that. If you are just relying on memorization, which is what 90% of you are going to be doing, even though I've told you multiple times, you need to understand it and apply it in ways that I have not already done for you, right? Same kinds and styles of questions. 99% of the time, when you get your 60% on this first exam, if I go over every single question on that exam, every one that you got wrong, which will be, if it's 40% of the questions that you got wrong, 20 questions out of the 50. If you get 20 questions wrong out of those 50, that's a 60%. You get 20 of them wrong, those 20 questions that you got wrong, that I'll go over the question, you will feel like an absolute fool for not seeing the right answer. 90% of the time, that's the case. Every time that I've gone over my exams with people. That means you're not applying the material that you probably already understood. Because when I go over the questions, people are looking at me and they're like, oh my God, you're right. We went over that critical thinking question in class about the diarrhea, right? So we talked about that. Like I gave you the example, the guy got sick. They found endospores in his poop, basically. So what caused it? The answer was easy because we just talked about endospores, but I put a bunch of those types of questions on your test. And suddenly, we haven't gone over endospores immediately right beforehand. And suddenly you guys are all picking salmonella. Facts. This happens every semester. So I took the time to go over endospores right beforehand. And hey, looks like you guys got it for a second there. You have to be able to carry that into the test. And so most of the time, people in the history of all of my other classes always picked salmonella. Almost all of my students did. Y'all picked Bacillus cereus this time. Why? Because we just went over endospores being made by Bacillus and Clostridium. You tell me right now, is that on your study sheet? It is. This is what I'm talking about. Be prepared for that. I'm not going to nitpick little details that aren't on your study sheet. You need to understand the concepts that are on the study sheet in big picture ways. But if there are little details like that, like if it's on the study sheet and it says that Bacillus and Clostridium make endospores, then you need to know that, period, because that will appear on the test. And so be prepared for that stuff. And I don't know what else to say about it. Obviously you can see that I get like worked up about it because I get ready. You're going to see, I'm going to tell you guys about this and drill it into your heads all this time right now, this morning. And those of you guys that are watching this afterwards in the recording, or that I've said it millions of times in the class, the same stuff. And I'm going to try to warn you guys that you need to be able to understand and apply. And then you're going to take the test and you're going to get the same 60% that everybody else does. And then I'm going to be going over the questions that you missed. And you're going to be feeling the same way that everybody else does, which is just that I did know this. I don't know why I couldn't find the right answer on the test. I don't know why either. But all I can do is tell you that I'm going to test you over that stuff. I'm going to test you over application and understanding and the ability to problem solve. That is why the first test is worth 5% of your grade and why the rest of the tests are worth more. Because you're going to take this first test and everybody always does the worst on the first test. And then now, after having gone through that experience, you will understand what's expected of you on the exams. Because people, I think they just don't believe me that it's going to be hard or something. Or you're preparing for the easier tests that you've taken in the past in other classes. You've prepared for other classes where in chemistry, where your questions are pulled directly from your quizzes. That is not this class. You've been preparing for easy classes, classes that have handed you your questions and answers. That is not this class. And nursing school will not be that class either. Nursing school will not hand you your questions and answers. Nursing school will hand you objectives that you have to learn, just like my study sheet, objectives that you have to learn. And then they will give you exams that have every single answer that is on your multiple choice will be correct. All of them will be correct answers. You just have to pick the most correct one. I'm not kidding. I'm not kidding. I'm not exaggerating. Please take this seriously. I'm not setting you up for failure with these tests. I'm setting you up to prepare for a harder class that you'll be taking next. And if you're not going into nursing school and you're going somewhere else, congratulations. It's not different. It's still going to be harder. This is not a class that they give you. This is the hardest class that you're ever going to take in the rest of your life. And then you go on to the future classes that depend on you having passed this class and they're going to be easier. That's not how life works. And so just take it seriously, please, when I'm telling you guys that you have to actually be able to understand and apply this stuff. Understanding and applying means that you can go through the questions that are on the quizzes or that we review in the reviews or that are in the Kahoots and whatever, and not just take the question and get the right answer, but you be able to understand why that right answer is right and why the wrong ones, not just by saying like, because it's the definition of it, but be able to actually explain in detail and go in depth as to why, and be able to use your own words to explain it out loud. That's more like what I'm talking about, okay? So I just want to get that out of the way because 60 is the average on my first test. It depends on what section you're in too. So my hybrid section usually does about a 56. They did, I think, 56 last semester. And my traditional section, my Monday, Wednesday AM typically does better than everybody else. I don't know why. I don't know why. It's just the type of people that tend to take that section for some reason. But my Monday, Wednesday AMs always do the best. And I think their average last semester was somewhere around like a 75 to 77. So when you guys are going through the Kahoots, when you're clicking through them individually, it should be giving you the answers when you're taking it, right? I mean, I'm pretty sure that's how it works. And if you want to retake the Kahoot, you can. I believe you can still retake it, but you'll just have to use a different username. So you could go back. I think you can just go back and click on that same link that's on the chapter stuff. It's the same link that we use in class in like the weekly thing. So if you go to like week 3, yeah, I think you can use a different username and go back through it. Am I right? Has anybody tried that? Am I right or wrong about that? I know we only have 7 people on here, but has anybody tried to go back through the Kahoots yet? I changed the format this semester, so that's why I'm asking. I can't take it as a student, so I can't see what you guys see. Anyways, hopefully somebody will have an answer to that, but you should be able to just go back through it in a different username. Yeah, new email, it says. So I'm not saying you guys have to create a whole new email address. I used to set it up where it was just username based and it didn't depend on your email. So yeah, you'll have to use a different username for that. Anyways, so moving on. I'm sorry I got so caught up in the test, but I do want you guys are here, man. You showed up to do the extra stuff. I want you guys to be prepared, right? So hopefully the other people will watch this too and see that this is because we're at that time now. Next week is the test, like it's crunch time. So if you're actually trying to study and prepare for the test appropriately, hopefully you've already started studying, right? If you're waiting until, oh, well, I'm going to wait till the day before the test, you will not be prepared for it, period. I'm sorry. It might have been good enough for other classes, but you won't be prepared for my class if you're waiting till the day before to study. I'm just saying. Like I said, my average, I'm scared and not prepared. Yeah, at least you're admitting it. Like this is the this is the start. So start working on the studying for that. Even if it is you're just doing some flashcards, at least you're getting something started now and not just waiting till the end. Working through the Kahoots again is fine. Having ChatGPT make you new questions is even better. How long are you allowed for the exam? The whole class period, so 75 minutes. So Brandy, you're in my hybrid group, so yeah, I can understand the confusion there. But yeah, it'll be 75 minutes, the typical lecture period. I know, and that's the thing is a lot of my students will be like, I don't know if I'm overstudying or understudying. Am I studying too much? Students that are very, very concerned about it and have been studying, and they're like, I've been studying and I still don't feel prepared. You are probably overstudying a little bit, but it's hard, and I get that. It's hard for me to explain it to you without just giving you the test, and that is literally why I make this test the least of your grade for all the tests. It's actually the least of your grade of any given block of things, the lowest amount that's a block of thing, like all of your in lab assignments that you guys upload, it's only worth 5% overall for all of them. So this one test, your unit one exam, is worth 5% of your grade. That's a lot for just one thing, but for an exam, it's the least of all of them. And so that way, when you guys know what to expect after unit one, everybody does better on unit two, even though it is way more complicated information. It is a considerably more difficult content, but everybody does better on unit two. Everybody does unit three even better, and everybody does the final the best, always. So, so I mean, and then I have a few students that will not always align with that, but overall on my averages, my class averages will reflect that very easily. In my online group, they usually don't put forth the effort that I see in my traditional groups because they're not being forced to go to class every day. And they just don't watch the lectures typically and don't put forth the effort to retain the information. They think that they can basically just look at summaries or skim through things and then that'll get them through. And that shows, that reflects in their data. Like I said, last semester, my hybrid, their first exam was around a 56 average, whereas my traditional classes, they were rocking about like a 72 to 75, depending on the section. That's more than a letter grade. It reflects that way the entire semester for my hybrid class. So if you're in the hybrid class, as long as you are keeping up with your work and you're actually watching the lectures and taking appropriate notes on the lectures and working to understand the material the same way as how our traditional groups are. Like if you're actually watching my traditional lecture recordings and taking notes along with that, and then being thoughtful when you're completing your Kahoots and your quizzes, you're going to get the same experience as my typical traditional classes, and you should be prepared. But I'm telling you that most of my hybrid students don't even watch the lectures. So I'm telling you that they don't. So most of them don't. And if you're one of those people like, I watched a couple of them, then you are one of the problem. Okay, so I'm just letting you guys out there know that. My traditional classes go to every lecture unless they're sick, and then it's their responsibility to watch it. I don't know what you're doing if you're watching it or not, but my traditional classes have to come to class. They go to every lecture. I don't know what happens that makes the hybrid people think that they're above that, but most of them do think that. So don't be that. Don't be that statistic. But we need to move on. I don't want to keep you guys on here too late. If you guys have any more questions about the exam stuff, we are having, remember, Monday and Tuesday is like review day, and you guys have plenty of time for questions about that sort of stuff as well. And we'll try to go over our critical thinking questions on Monday and Tuesday, and I'll have those recordings posted for my online folks. So if you are hybrid, that those will be posted for you. as well, just like our traditional recording for stuff will. And I'm scaring you, if I feel like I'm scaring you guys or scaremongering it a little bit, that kind of is on purpose, because 60% average is pretty low, but that's the average for everybody. And it's been getting better every semester because I've been trying more and more to be sure that I'm preparing you guys and making sure that everybody's aware. So every semester it gets better. And that hybrid class just really likes to hold on to that lower grade. And there's not a lot I can do to force people to prepare. So all I can do is just warn you guys. Just do what you need to do to learn the material, right? And I try to give you as many resources as I possibly humanly can to help you with that learning. If you're having a hard time understanding the difference between gram positive and gram negative, go listen to the audio short about it. You don't want to rewatch the whole lecture? That's great. Or you watch the lecture, but you want just to focus on that one topic, go listen to the audio short. Have ChatGPT write you a summary for it. Go look in the notes and reread them and rewrite them in your own words. Go back to the textbook and see what the textbook has to say and all that stuff. You can work with other people, create Kahoots with people, create Quizlets, new Quizlets with people, but beating a dead horse by going over the same questions over and over and over and over again, and just making sure you know the right answer isn't going to help if you don't understand why, right? That's my point there. So let's move on for real this time. Okay, I promise. And then we're going to knock out this other stuff. So for chapter 5, trophozoites versus cysts, remember our trophozoites are our active feeding. Troph means eat. And if you are eating, you must be active. You must be doing things, because eating means you're getting energy and nutrients to make, new cell parts and things like that. So trophozoites are active, motile, feeding, reproductive stage. Cysts are dormant and resistant, and they're adapted for harsh conditions. That sounds like an endospore to you. Congratulations, you're starting to make that correlation. So endospores that are made by bacteria are analogous or similar to cysts that are made by protozoa. The vegetative forms of bacteria are active and free feeding stage of those bacteria. They're living, they're dividing. Vegetative bacteria are analogous to trophozoites for the protozoa. All right. Tigers fight, cats cover. I mean, what I want to say about this one, trophozoites are functional. So tigers fight, trophozoites are functional. And then cysts are adapted for taking cover or hiding. Okay, so I don't know if that helps you remember that, but that was my it made more sense to me when I wrote it. That's how I remembered it. All right, so our questions regarding this. The parasitology lab is examining a patient's stool and identifies both trophozoites and cysts. Okay, so we are seeing trophozoites and cysts of protozoa in somebody's poop. So trophozoites, those are our active. And our cysts equal our dormant. And that's how we should be thinking about that when we start approaching this question. So already I'm breaking this question down again. So they're experiencing GI symptoms such as diarrhea and abdominal pain. All right. We know we have trophozoites in there, and we know we have cysts in there. And this person's having diarrhea and pain. Based on the fact that we have trophozoites and cysts there, which of the following statements best explains their roles in the infection cycle? Okay, so like what are they doing in the infection cycle that's leading to the infection presentation? Right? So if our trophozoites are the active forms and the cysts are the dormant forms, let's go through our choices. Trophozoites are responsible for causing symptoms. I would imagine they could be, because they're the active form in the host due to their active feeding. Right? Okay, yeah, that's aligning with what we're saying. This is driving me insane. While the cysts help in transmitting the protozoan to new hosts through contamination. This makes sense because the cysts are the dormant form, so they'll survive the environment and get into the host, but then turn into the trophozoites, the active form. And that's what's causing the actual active feeding, producing toxins and everything like that, where we have the actual symptoms. So Choice A is looking pretty good so far, but we want to check out our other choices. Trophozoites are primarily involved in survival and transmission, whereas the cysts are involved in causing the infection. Cysts, so in order for, what is infection first? Infection means that an organism is populating itself inside of the host where it shouldn't be and in numbers that it shouldn't be at, right? So it's establishing itself in an inside of a host environment of some kind where it shouldn't be, right? So can a cyst do that? No, because it can't even multiply. It's dormant, it's inert, it's not doing anything, it does nothing. It just is waiting for it to turn into a trophozoite, which will do all of the work. So these roles are switched here in B. C says trophozoites and cysts both contribute equally to the infection and transmission of the protozoan by actively feeding and surviving. We've already said that the cysts don't actively feed. They'll survive, but they don't actively feed. Trophozoites will be limited in what conditions they can survive in. Maybe they can't survive in chlorinated water in a swimming pool, but the cyst can. But then you get the cyst inside of you, then it turns into the trophozoite, and that's what multiplies and causes disease. So it's not C. Trophozoites are dormant, and nope. Can we already say no on this, right? So trophozoites are not the dormant ones, they're the active ones. The cysts are the dormant ones. So already I don't even need to read the rest of that statement. It's wrong in the beginning. So the whole thing can't be right. So we can say that A is the correct one there. In an experimental study, researchers exposed protozoan cysts. So these are our dormant and hardy guys. Dormant, hardy survivors, right? So they can survive heat and desiccation, so being dried out. The study finds that the cysts survive these conditions, but trophozoites don't. What can be concluded about the function and adaptation of protozoan cysts? So are cysts adapted for active growth? This is just active growth. Oh my gosh. And nutrient absorption? No. Trophozoites, they survive harsh conditions, yes, but are they adapted for active growth? No. So this one's wrong already. Cysts are designed for survival. My back is killing me. Just a second. Cysts are designed for survival under adverse environmental conditions. Yes. Making them resistant to heat and desiccation. Yes. And are trophozoites less resilient? Yeah. So B is looking pretty good. Cysts are more susceptible to the environmental stressors compared to trophozoites, which are, that's not going to be true, right? So they're not more susceptible. Susceptible is like sensitive, right? So cysts are more resistant compared to trophozoites. Trophozoites are not more durable. So this is not right. Cysts are involved in active reproduction. Y'all, no. So B is our answer. Cysts are dormant. The trophozoites are reproductive. Okay. Chapter 7, one of the most missed concepts, which we're almost there. Chapter 7, one of our most missed concepts is understanding what a cytopathic effect is. I am just drawn right through this word. A cytopathic effect is basically anything that you would see structurally or functionally changed in the host cell as a result of that viral infection. So as a researcher, as a lab assistant that will be looking at this, I know that you guys aren't going to be doing that lab stuff, but if you were, what's happening in the lab to your patient samples? They can look at your patient's like tissue sample or something like that and see that your cells are having structural or functional changes in the microscope. And those are called cytopathic effects. They happen because when a virus is in a cell, it takes over the cell's machinery, so disrupts normal cell function. They also will put their spikes on the surface of the cell that can affect that cell's ability to attach to surfaces, its ability to orient itself, like if it's supposed to be upright or downright based on the structures of the cell. Its ability to know when it's supposed to be replicating versus not replicating, and all of those things can be affected. We've already talked about syncytia, whenever we have cells sort of merging together as a result of the infection. and the spikes on the surface and everything, causing cells to merge together to make giant multi-cell single cells, basically containing all the contents of multiple cells. It can cause our cells to lyse or burst open. Our cells can swell just from all the stuff that's building up in them. We have the development of these things called inclusion bodies, where our cells are accumulating viral parts, and we're trying to basically put them somewhere that they belong in the cell and make them go somewhere and consolidate them there. And that's what an inclusion body would be. But seeing any of these effects is a cytopathic effect there. So sick cells develop crazy alterations, and here these alterations are we're seeing that's our cytopathic effects. Cytopathic means cell, path means disease, and effect. So cell disease effects. That's what that word means. All right, which of the following best describes or best exhibits a cytopathic effect in the context of a viral infection? So with cytopathic effects, we're looking at effects of the cell that we would be able to directly observe, right? So a viral protein inhibits host cell replication. How would you know by looking at the cell that the cell is not replicating? Just by looking at it. That's going to be difficult. Structural and functional changes observed in our host cells. This is very on the nose, right? So we actually said in our previous slide that a cytopathic effect is literally a structural and functional change that we'll observe in the cell. And here, this is very, very literal version of that. So this is going to be our answer, kind of obviously, it's a direct usage of that terminology. But also, if we go through our other choices, we'll see. C, the process by which viruses are released from the cells. This is not a cytopathic effect. That's just how the virus is released from the cell. That might not look anything weird or change the cell somehow or whatever. The actual process by which they're getting released is not a cytopathic effect. It's just how the viruses are getting released. And we have the ability of viruses to integrate into the host cell genome. Now, that's something that we don't want to have happen. We don't want viruses putting their genomes into ours and becoming pro-viruses. We don't want to see that. But is that a cytopathic effect? Is that something we can see causing disease to that cell? It is not. So B is going to be our answer here. So we're looking for actual structural or functional observable effects on the cell. These other things might not be ideal, but they are not observable effects. All right, a lab technician observes that cells are having fused from multinucleated giant cells that exhibit severe swelling. So here we've got two things going on. We've got syncytia and we've got swelling that have both been defined to us as cytopathic effects. And now this is just one of those moments where you have to remember what a syncytium is. And that is defined as whenever we have those cells merging together to create those giant cells, right? So which of the following viral infections is most likely associated with these effects based on their name alone? So we're looking at maybe viruses that have in their name swelling or syncytia, right? So what are our choices? Herpes simplex virus doesn't indicate anything to me about swelling or syncytia. Influenza doesn't directly to me. Human immunodeficiency virus suggests that we would have immune problems, but it doesn't hint at syncytia or swelling. And then very again on the nose here, this one's a pretty obvious one, but syncytial. It says it in the name that we're going to create a cytopathic effect. Okay. And so remember that syncytia are a cytopathic effect. All right, so let's talk about our lytic cycle versus the lysogenic cycle and lysogenic conversion. This is a big one for people, right? Because when you are a phage, which I'm going to abbreviate phage as this, because it's just easier, and that's typically what we do. Oh my God. It's what we do in science with these guys. So it's like phi, right? So this is, yeah. But I can, yeah. What are you doing, computer? Can you work with me here? Jeez. Let's get rid of that. God, I'd love to just go back to my mouse. Beige. Anyways, oh my gosh, why? I don't understand. I'm not doing anything weird. I promise I'm not. I'm just writing. Anyways, it's a five. So basically, I guess we're not going to use that symbol. So phages. Goodness, I don't understand. All right, so a phage. It can go through some phages. I want to be very clear about this. Will only go through the lytic cycle. So sometimes they'll only be lytic. And what is lytic? That's making copies of the virus. Viral reproduction. That's what the lytic cycle is. Okay. Two, you can't have phages that will switch between lytic and lysogenic. Okay. So they'll switch between these. So lytic is making virus. That's an arrow, I promise. And then lysogenic is whenever you are hiding phage DNA in bacterial genome. If you can switch between these, then you are called a temperate phage. Okay. That's phage. We'll try again. No, okay, forget it. Forget it. It's driving me insane. Anyways, God, I don't know, man. This tablet, and I'm like, why did I ever buy it if it's just gonna do that? All right, so anyways, so when you get this situation of having lysogenic, sometimes it's gen, and that's the part where I remember the difference because lyso also kind of hints there should be lysis going on. There is not. Lytic phase, we have the actual copies of the virus being made until the bacteria explodes via lysis. Okay. Lysogenic, please focus on the genes. We're putting viral genes into the bacterial genes. So lysogenic. Okay. So the lysogenic phase, where we put the viral genome into the bacterial genome, it becomes the prophage. Temperate phages can switch between those. During the lysogenic cycle, if we are incorporating viral DNA into bacterial DNA, sometimes we can also add in traits that help with survivability of the bacteria. Things like toxin production and antibiotic resistance. We're making additional structures in the cell. Stuff like that can be carried within the virus genome. And then when it's incorporated into the bacterial genome, give them extra traits. That is called lysogenic conversion. Here. So lysogenic conversion. You can tell I'm just having a great time with this new tablet. I don't know what causes it to do what it does. So lysogenic conversion, it refers to the change in the host cell's properties. So in this case, the host cell is the bacteria as a result of the presence of that prophage DNA. Okay, so that, like I said, we could be making toxins. We have down here some examples. Toxins like diphtheria toxin produced by the bacterium. Thank goodness I'm not making you spell this one out. Corynebacterium diphtheriae. Okay, so that bacteria, Corynebacterium diphtheriae, they produce this toxin that allows the development of the disease that we know as diphtheria. Now, we don't have diphtheria here in the United States really anymore, but what it does is it allows the bacteria to create this sort of pseudo membrane in the throat that can entirely block breathing. It can also lead to development of pharyngitis, coughing, and other upper respiratory and even mid to lower, excuse me, this on silent, mid to lower respiratory tract infections. Okay. And that's what that is. We won't really be talking too much about diphtheria in this course, but it is a disease to be aware of. It does affect other, nations that are less advanced maybe than ours is technically. So lively lions instantly take control, but lazy leopards lie in wait. So here is my little explanation for my little memory tool for this one. Lytic, lysis, we'll have actually breaking open of a cell. So the lively lions were lively because we're in that lytic phase, and that will lead to the lysis, lively, lion, lie. Okay, so lively, lysis, that's our lively lions. Okay, the lively lions instantly take control, so they're starting instantly going to be making copies of the virus. Okay, so our lazy leopards, or lazy because we're kind of in a resting or waiting state. And so we're basically going to lie in wait because we are waiting for us to make copies, and we're just waiting in the genome is where we're waiting. So that's basically why we'll have that divided up that way. We're almost there. Okay, here's our questions regarding lysogeny and the lytic phase. We have, which of the following best describes the main difference between lytic and lysogenic cycles? The lytic cycle involves the integration of phage DNA. That's not right, because we'll make our notes off to the side first. So I do recommend doing this on your scratch paper when you guys are doing the test. So if the question's already asking, what's the difference between lytic and lysogenic cycles? So this is what I would be considering kind of that second level of questions. We have our first level of questions, which is our basic definition and term. And this is just a step above that because I'm just expecting you to compare kind of two terms, lytic and lysogenic at the same time. So we're going to have to find an answer that kind of addresses both at the same time. So it's like our second level. And then our third level of question asking would be our more critical thinking or direct, true application questions, right? So what best describes the main difference between these guys? So we know lytic, this is when we're making our virus copies. So this is our reproduction phase. And then our lysogenic, this is a G. This is going to be a phase where we have our prophage DNA inserted in the bacterial DNA and hiding out or waiting, right? So that's our little note off to the side when we're going to choose our choices. So that's how we can knock out number one, because lytic, it starts off, lytic involves integration of the phage DNA into the host genome. That's not lytic, that's lysogenic. Next choice, the lytic cycle results in the destruction of the host cell versus through lysis. That is what happens in a reproductive stage. While the lysogenic cycle involves integration of the phage DNA into the host genome without immediate death of the cell. And that's true too. So it's looking like B is probably our answer. We have no reason to think it wouldn't be, but let's look at our other choices and make sure there's not a better choice. All right, the C, the lytic cycle occurs in all types of bacteria. This is true, while the lysogenic cycle only occurs, and this is where it's going to go south. The lysogenic cycle can only occur in temperate bacteria, but are gram-positives the only ones that can do that? We did not mention that at all, so that is not something that we talked about. It's not a truth either, it's falsehoods. So we did, that's not, nope. Just knock it out. You don't have to be gram-positive to be temperate phage. That doesn't need to be. So phages aren't gram-positive or gram-negative, right? The bacteria is gram-positive or gram-negative. Then with your phage and you are infecting certain kinds of bacteria, whether it's gram-positive or gram-negative, doesn't affect whether you are going to be temperate or not. What kind of phage you are depends on whether you're going to be able to go from lysogenic and lytic and cycle between the two. What type of phage you are determines that, not what type of bacteria you're infecting. All right, the lysogenic cycle produces new phage particles immediately, where the lytic cycle does not produce phages. And this is kind of the opposite of that. So B is still our choice. Our next question. So you see what I'm talking about here. We could easily just like try to find the right answer and move on, but understanding the reasons why these other guys are wrong can also help us understand the topic a little bit better. All right, the patient presents with a bacterial infection caused by Corynebacterium diphtheriae. Here we are again. We just mentioned it in the previous slide, but even if I don't expect you to know Corynebacterium diphtheriae, and I might do this to you on the test. I might mention a bacterial strain that I never mentioned previously in our talk. But if I do that, I will give you all of the information that you need about it to answer the question. So here we have a bacterium. So I'm just going to, we have some kind of bacteria. We show that the strain is producing some kind of toxin. And based on what we know about lysogeny, so lysogeny tells us this stuff over here, that the prophage DNA is inserted, and that's how the phage is hiding. And we know about lysogeny that that can sometimes lead to that term that we called lysogenic conversion. So this sometimes happens with lysogeny, where we will gain traits because of it, because of the lysogeny. The bacteria will gain traits because of the viral infection. Okay, so what are our choices? The bacterial strain is carrying a prophage, that's a lining, that has integrated into its own genome, and that prophage encodes a toxin. That pretty much describes exactly like what you would see with lysogenic conversion as a result of that. So this is probably our choice, but let's go through the other ones. The bacterium has a high mutation rate that causes it to produce toxins. What does that have to do with lysogeny? How does lysogeny tell you about it? It literally says we need to know about lysogeny to answer the question. Okay, so this is not relating to that. The bacterial's ability to mutate has nothing to do with this. The bacteria has a high resistant to antibiotics. Because it's resistant to antibiotics, it makes toxins. This actually makes literally no sense at all. And we'll learn more about why that is when we learn about antibiotics and why bacteria make toxins. But like usually if you're making a toxin, that's an exotoxin that you're secreting from the cell. How does that relate to your ability to be resistant to a drug that kills bacteria? I'm not really sure. This is a stretch. But it definitely doesn't have to do with lysogeny by a virus, does it? So then we have D, the infection has caused the bacteria to undergo genetic recombination, leading to toxin production. So genetic recombination is really telling us that we have shifted our own genes around as a result of being infected by this virus. And that isn't really what's happening with lysogeny. Lysogeny, you're just inserting new genes in with your viral genome. And that's not causing us to go genetic recombination necessarily. That's going to lead to toxin production. More specifically, this is a very sort of weird roundabout way to almost talk about what we're getting into. What's the most correct answer here? The bacterial strain has a prophage in it that leads to the toxin being produced. This is the most correct answer. We can almost, this is a tricky one because we can almost include D. Almost. Because we are changing our genetic information, but this is A is the most correct here. Right. All right. So moving on, last thing, this is the last one. Prions, misfolded proteins that when they are misfolded, they don't interact with the environment well anymore. They stick together, causing holes in the tissue and then leading to sponge-like brain tissues. Usually affects the brain. Sponge-like brain tissue leading to disease development in the brain, and that is why we call it spongiform encephalopathy. And so a spongiform, sponge-like, encephalopathy, encephalo means brain, pathy means disease. This is a lot of information that sort of breaks down prions and how they work and examples of these diseases. But for sure, for this class, and I believe that it is mentioned in that study sheet, the example of the prion disease for humans, it's called Creutzfeldt-Jakob disease. It's this word here, CJD, Creutzfeldt-Jakob. Again, you won't have to spell it, but you do have to recognize it on a test as that being the disease of a prion disease is found in humans. So I'm fidgeting and I should probably be holding a fidget spinner or something instead of just rubbing my eyes and my face all the time. But I don't have a fidget spinner. What kind of travesty is this? So I'll just grab this thing instead. Anyways, Creutzfeldt-Jakob disease is a prion disease of humans. So that's like the biggie for humans as far as the prion disease caused by misfolded proteins and all that. So we're going to leave it at that. But here is our little mnemonic prion D. Problems really involve only nerve damage. So really our nervous system is the only place that we see the expression of these diseases, and that these misfolded proteins are going to be leading to those problems in those nervous systems. It's just because of the inability of that nervous system to heal itself, as well as its sort of secluded nature, as far as we don't have the same immune responses there and things like that. All right, so which of the following statements accurately describes prions? Before we go with our choices, we know that prions are misfolded proteins and these guys basically, I don't want to say coagulate, but they clump together and that causes brain diseases. All right, that's our watered down note for this question. So prions. Prions are viruses. They are not viruses. They are misfolded proteins. So we can move on to the next question, the next choice. Prions are misfolded proteins. Okay, they are misfolded proteins. Do these misfolded proteins induce other normal proteins to misfold? They do. So it's like a cascade effect. Normal protein misfolds and then causes other normal proteins to misfold. And then they all like clump together and they cause brain disease as a result of that. So B is probably our choice. C says prions are bacteria that release toxins. Prions are not bacteria, they're misfolded proteins. And D says prions are fungi that cause infection by spreading spores. Prions are not fungi, they are misfolded proteins. So B, obviously, this one's not too bad for this question. Again, another pretty decent example of like that type 2 kind of question. I'm not asking for too much critical thinking here. A little bit of just understanding goes a long way in these types of questions. All right, question 2 for this. A 55-year-old patient presents with rapid onset dementia, coordination problems, and muscle twitching. So we've got some nervous system issues here. Diagnostic tests indicate abnormal prion proteins. So we have our misfolded proteins that are going to clump together to cause brain diseases here. Which of the following diseases is most likely associated with these findings? Especially for our course, which disease did we just talk about that is a prion disease? It's C. So this is a matter of, again, kind of a type 2 question. I don't consider this a critical thinking question because it's kind of basically just asking you which of the following is a prion disease that we learned about in our class. So all these other diseases can be related to neurological degeneration, but we know for sure that Creutzfeldt-Jakob disease is a true prion disease where I've introduced into the body a misfolded protein that then caused other proteins to misfold. It's infectious. It can spread disease. It's contagious. With Alzheimer's disease, do we have misfolded proteins? Amyloid proteins is what we call them in Alzheimer's disease. Now, we didn't talk about it in our class, but as it turns out, yes, they are. Alzheimer's disease, it is caused by something very similar to prion diseases. It's proteins that are misfolding and then causing damage to brain tissue whenever they're sticking together and basically clumping together and causing damage to the brain tissue. That sounds a lot like prion disease, but we didn't introduce the misfolded proteins. These guys just misfold because of old age. It just happens. And it's not transmissible. You can't catch Alzheimer's from somebody else from eating their brain tissue or something like that. We've never seen any incidents of that. But prion diseases are transmissible and they are caused by introducing a misfolded protein and causing your normal proteins to misfold as a result. So there's a line there. Prion diseases are transmissible, can be spread from person to person. These other guys cannot be. There's been no documented case of it. Now, genetically spread is not the same thing, right? If it's in your genetics, it was in your genetics when you were born, you're encoded for it. That is not transmitted, right? So it's not transmitted horizontally. Horizontal transmission is from person to person. Okay. Yeah, thanks for joining us today, Lakita. And that is the end. So thanks for joining us and thanks for sticking around for as long as you guys did. I know we went over time. We took some more time, but we were able to talk a little bit more about the yeah, we went a lot over time. I usually don't like to go this long, but thank you for letting me talk about the exam. I hope I answered some of your questions about the exam. And I know you're not feeling any better about it. That's okay. You're going to be nervous. That's normal. Even me being the most prepared I've ever been for any of my exams in my entire life, and and getting hundreds on my exams and doing perfectly. Everybody, you're going to go in nervous. That part's never going to go away. Never, even when you're the most prepared you could possibly be. I've never gone into any test in my entire life. Even if it was my ABCs, I'd probably be nervous about that test. So keep that in mind. It's normal to be nervous. It's normal to be having that anxiety about it, and that's okay. And we'll go over some little tricks that can hopefully help with that anxiety on test day when we get there. So everybody, good luck with your studying, and I will see you on Monday and Tuesday for our review stuff. Monday, for Monday, Wednesday people, we will be doing the games for prepping for the exam for Wednesday. And here, my Tuesday, Thursday people will have a game on Wednesday to prep for the exam for Thursday. So everybody be ready for whichever day you're going to prepare. Come join us for our games. You have the Zoom link. It's always the same Zoom link as even this one is. So I'll see you guys then. Anybody have any questions? If you do, now's the time. Thank you guys for coming. Have a good weekend. Get ready for gram stains on Monday and Tuesday. I'm excited. Thanks, guys. All right, and I'm going to go ahead and cut it off here.